Hello, this is Ed Chapman, and this video cast is going to cover the function of enzymes. Remember, enzymes are a certain type of proteins, and proteins that actually do things or cause things to happen are what we call enzymes. All right, enzymes are what we call biological catalysts, and a catalyst is a material that causes a chemical reaction to happen faster. So you can think of enzymes as biological catalysts. Okay, that's an important word. They make reactions happen faster than they would if the enzyme wasn't present. And the cool thing about enzymes and catalysts in general is they're not consumed by the chemical reaction. They're used over and over again. They're catalysts. They make the reaction happen, but they're not actually part of the reaction themselves. Now, enzymes are absolutely necessary for the metabolism or the functioning of the chemistry inside of all living cells. There are basically two different categories of metabolic processes inside of cells. There is the digestive, or the act of taking polymers and breaking them down into monomers. And then there is synthesis, which is just the opposite. Synthesis, of course, builds polymers by hooking together or bonding together monomers. So these are opposite types of chemistry. And enzymes are critical for both of these types of metabolic processes. Now, I like to think of digestion as breaking and synthesis as building. So if we use an analogy here of scissors and staplers, I'm hoping you can see that a pair of scissors is a breaking tool. It's a tool that takes things apart, whereas a stapler is a building tool. It puts things together. So you can think of enzymes as enzymes that act as staplers or scissors. Okay? The staplers are the synthetic enzymes or the synthesizer enzymes, and the scissor enzymes would be like the digestive enzymes. One of the ways you can visualize how an enzyme works is using diagrams like this, which have things that fit together like puzzle pieces. And what makes jigsaw puzzles fun is that only certain pieces will fit together. And in the case of enzymes, enzymes have a location in their three-dimensional structure called the active site. So what I've just outlined here is the active site. And it's the place where another molecule can fit. Okay, now the molecule that fits into an enzyme is called its substrate. All right, and this is a very specific interaction here. Only certain substrates can fit into each enzyme. And when they fit together, we end up with something called an enzyme-substrate complex. And this is where the chemistry happens. By fitting into the enzyme, the substrate is actually changed into a product. And in this example, we have a large substrate molecule being broken down into two products. So you could call this a digestive enzyme. Okay? Now here's a little bit more specific example. We have sucrose, which is a double sugar or disaccharide. Remember, sucrose is a glucose attached to a fructose by a bond here that was created by dehydration synthesis. And here we have an enzyme that's found in your digestive system called sucrase. Now, sucrase has an active site that can receive a sucrose molecule. They'll fit together. So here you see them fit together. And what happens is the sucrase enzyme is able to break this bond and allow the glucose molecule to separate from the fructose molecule. So this is an example of digestion. Okay, so sucrase is a digestive enzyme. It's kind of like the scissors, okay, because it clips that bond, breaking the glucose apart from the fructose. And so these would be the products. All right. Now, all enzymes have environments where they work the best. And the two abiotic factors that are most important for enzyme function are pH and temperature. Okay? So if we make a graph of how well an enzyme works, okay, the rate of the reaction. So as we go up here, the enzyme is working faster and faster. Okay? And as we go down, it's working slower. All right? Here we have the graph of some data for an enzyme. Let's say this is enzyme number 1, right? And you'll notice this enzyme doesn't work very well at 0 pH or 1 pH. But when we get to about a pH of 2, okay, the enzyme is working very well. So this is its optimum. Okay, so enzyme number 1's optimum is a pH of about 2. So number 1 works best at pH equals 2. All right, this might be an enzyme that works in your stomach, for example, where the pH is very low. Now, enzyme number 2 over here, represented by this red line, it's not hardly doing anything at low pH. When we start getting up close to neutral, okay, maybe a little bit past, okay, that's where enzyme number 2 is working the best. Okay, that's where it's working the fastest. And it's working the fastest at a pH that's 7. So number 2 works best at pH equals 7. So this could be an enzyme here that has evolved to do its job in a place where pH is close to neutral, like maybe in your mouth. This could be a saliva enzyme, whereas enzyme number 1 could be a stomach enzyme. So do you see how pH can affect how well an enzyme works? That's the idea here. Now the other thing is temperature. Here we have 4 different enzyme, a graph just like we did before. Three different enzymes here at different temperatures in Celsius or centigrade. So this is 4 degrees Celsius, 37 degrees Celsius, and 95 degrees Celsius. Okay, and look at enzyme number 1, enzyme number 2, and enzyme number 3. Okay, so you can immediately see that enzyme number 1 works really well at 4 degrees Celsius. That's very cold. Okay, so this might be an enzyme that's working inside the body of, like, say, a fish that lives in Arctic waters and very cold water. This enzyme here, number 2, is working its best at 37 degrees, which is close to human body temperature. So this could be like an enzyme in your blood or something. And finally, this enzyme here, represented by the green line, number 3, it's peaking in its efficiency or its ability to work at 95 degrees Celsius, which is really close to boiling. This could be an enzyme found inside of a bacteria that lives in hot springs or inside of geysers or something like that. All right, so I want you to see here from this graph that temperature also affects how well an enzyme works. All right, thank you for listening. That's the end of the video cast.